Hello. In this section of our studies of anatomy and physiology, we're going to spend time studying the cardiovascular system. And you will see that it is not as complicated as it may look like at the beginning of your studies. Naturally, it's going to take some time for you to become more comfortable, to grasp the new terminology which is to be applied in this section of your studies. And in total, when we come towards the end, which will be completed with physiology of cardiovascular system, I hope that you would be able to perhaps go back, revisit a couple of chapters from this recording, and perhaps seek for some additional clarification. So let's not waste too much time. Let's go straight into the main topic. And for that reason, I need to share the screen, which is going to be done in a moment. And I need to open up the slideshow to accompany our discussion. So we're going to go with the first slide. And the purpose of this is to practically make you see the big picture, so to say, of what the cardiovascular system looks like. If you would be asked this question before even reading a first paragraph, I'm sure you would come up right away with the idea cardio, heart, vascular, blood vessels must be then composed of several components, of which three, the most important ones, are the heart itself, which is acting as a very powerful pump. And then the rest of the system is to be composed of multiple arteries and veins. Let's make sure that we understand immediately what the term artery actually denotes. Artery is a blood vessel which is taking away blood from the heart. So on this illustration, we have here one artery, which is the largest continuous blood vessel in the entire body. Its name is aorta. And you notice right away that it is colored red. Definitely the blood goes away from the heart and it keeps going into multiple regions of the body. However, there is another blood vessel which is colored blue, and its name is pulmonary trunk. It also takes blood away from the heart. However, this time it diverts the blood into lungs for the purpose of oxygenation. So, in other words, the color difference which has been applied to this diagram is not of any kind of peculiar value. Rather, we can explain it. Both vessels are taking blood away from the heart, with difference that aorta brings out of the heart's chambers oxygenated blood, while pulmonary trunk is doing the same with the blood which is deoxygenated, and for that reason it has to be sent to lungs for the purpose of oxygenation. So if we follow what's happening on this diagram alone, we can also notice that the heart is also divided into four different chambers. Two of them that are located on the upper part of the heart, they're receiving chambers, and they're called atria. Singular is atrium, and you can see here we have the right atrium and the left atrium next to it. Again, the color difference between them denotes that the right atrium is the one which receives venous deoxygenated blood from the body, and then, as white color arrow indicates it, it sends it down from right atrium into right-sided ventricle, from which deoxygenated blood enters the pulmonary trunk. Left atrium and left ventricle are other two chambers, and left atrium also receives the blood, which comes this time through the pulmonary veins. Veins bring oxygenated blood back into left atrium, which pumps the blood into left ventricle, and upon a contraction of left ventricular musculature, blood is pumped out into aorta and through it distributed to all different parts of the body. So we're left now to try to identify and to define what a vein is. So a vein, regardless whether it carries oxygenated or deoxygenated blood is a blood vessel which brings blood back to the heart. So when the blood was oxygenated in lungs, basically from lungs, it's drained away by 4 pulmonary veins, 2 on the left, 2 on the right, and oxygenated blood flowing through pulmonary veins re-enters the left atrium of the heart. On the other side, we have 2 major so-called systemic veins, one coming from the upper parts of the body, namely the head, the neck, upper limbs, and its name is superior vena cava, superior hollow vein, which, upon collecting blood from these parts of the body, enters into right atrium. From the lower parts of the body, so anything that's distal to the diaphragm, we have another hollow vein, this time named inferior cava vein, which collects blood from lower limbs, pelvis, abdomen, and brings deoxygenated carbon dioxide-rich blood also into right atrium of the heart. And as you can see, in between different arteries and veins, regardless of their colors, we have interesting and very important network of pulmonary capillaries in both left and right lung. And then we have on the very top and the very bottom part of this diagram, we have so-called systemic capillaries of both upper and lower body. So truly, the system that we're talking about, the cardiovascular, is not composed only of heart, arteries, and veins, but at this time, we must include the idea that we also have practically immeasurable amount of capillaries, smallest caliber blood vessels, and their quality is that their walls are so thin that they enable exchange of gases, carbon dioxide for oxygen or vice versa, and also exchange of nutrients for waste products. Basically, capillaries are the most functional part of the cardiovascular system. However, because of their minute size, we actually do not study them in a regional manner. We just have to understand what they do and that they are part of practically any tissues within the living body. Let's go to the next diagram and let's try to immediately grasp another important thing which differentiates arteries from veins. I'm going to expand here a little bit and to try to so-called stir the pot by giving you some information which may not be necessarily related to what we do. However, that will explain the importance of understanding this part of your studies. When you compare on the upper part of the diagram what cross-section looks like if we cut open one typical peripheral artery, and then we do the same with approximately same size, similar caliber vein, it is right away noticeable that there is a difference the way they're built. So there are some similarities. Each of them, whether it's artery or vein, has its innermost layer, which is made of flat endothelial cells. That layer we call tunica interna. Its most important quality is that tunica interna actually creates a very smooth surface that allows very uninterrupted flow of blood without triggering, even involuntarily or accidentally, the pathway which would lead to blood clotting, to blood coagulation. Endothelium in arteries is perfectly smooth. However, endothelium in veins is going to be occasionally thrown into folds that are called valves. Valves are particularly present in veins of the lower limb because we have to offset the force of gravity. The gravitational force would naturally allow blood to pool down in the lowest parts of our lower limbs, in our feet, in our legs, around our ankles. But with this kind of simple yet very effective mechanical mechanism, we are practically preventing reversed, so-called retrograde flow of blood. That is innermost layer, tunica interna. The middle layer is the one which we also see with a huge difference between artery and a vein. And that will explain what I just said a couple of minutes ago. I'm going to throw at you certain information which may not look like it's directly connected. It's about blood pressure in a typical adult healthy person. We know that it is measured as 120 over 80 millimeters of mercury. That is the pressure that blood exerts against the wall of blood vessel. So to think about heart being as big as your own clenched fist, to be able to generate such a force to maintain the blood pressure at the peak of its contraction at 120 millimeters of mercury and at the peak of its relaxation to still maintain the pressure at 80 millimeters of mercury would be a little bit too much and too hard to imagine that structure as small as a heart is capable of doing this practically throughout our lifetime. This is where the middle layer or tunica media comes into effect. Obviously, it is made of smooth muscles. That's why we call it also tunica media or the muscularis, referring to its muscular structure, and being composed of smooth muscle fibers capable of producing pretty much long and extended contractile force. It is the tunica media in arteries which actually produces an extra contraction, pushing blood further away from the heart and being directly responsible for maintenance of so-called the diastolic pressure, the lower value of about 80 millimeters of mercury, that would be between two heart's contraction. If we compare the thickness of tunica media within a wall of an artery and immediately try to see how much of that we're seeing in a wall of a vein, we notice the huge difference. Arteries have much thicker tunica media. Why? They need to generate that pressure that is overcoming the tissue resistance. So blood forcefully has to be injected into tissue, and it cannot be done in a passive form. Blood, once it starts flowing out of the tissue into veins, basically is facing less and less resistance. Therefore, veins do not have to produce the same effect with their tunica media. Veins are quite passively filled, and blood just keeps flowing through a system of veins based on the pressure gradient. Finally, if we understood this last section about tunica media, there is a comparable outermost layer that is called tunica externa. It's pretty much wet, slippery coat, which prevents arteries or veins to be accidentally squished and compressed between any other two parts of the body, particularly muscles, when and if they do contraction. So both arteries and veins tend to slip away from a point of highest pressure, trying to just position themselves in a safe distance so that blood flow through artery or vein doesn't get interrupted. Compared to artery and vein and their wall, basically at the bottom of the screen, now you're seeing another illustration that is very important to understand. It's a typical diagram that illustrates what the wall of capillary looks like. And you can see right away, it only has one layer, simple single layer of endothelial cells, making smooth inner surface, enabling blood to flow without any resistance or turbulence. And these endothelial cells appear to be attached to a very simple basement membrane. So the walls of capillaries do not have muscularis, do not have external coat or external tunic. Basically, there are even smaller gaps between endothelial cells, and through these gaps, we're seeing opportunity for gases to keep going between blood and tissues, both directions, as well as nutrients and waste products that also travel both ways across the wall of capillary. So let's go quickly backwards to the previous diagram because now it's obvious. These are veins and these are arteries and these are capillaries. Do we have anything else that we have to add here to make it somewhat more complete description of what the cardiovascular system is made of. There is something that we need to add. So if we follow, let's say, aorta, it is large, big artery, which keeps branching. So it sends some branches up there to the head and neck. Then it sends some branches to upper limbs. Then it keeps sending some branches to walls of the thorax or chest and abdominopelvic cavity. And finally, it sends some branches that are still called arteries into our lower limbs. These arteries keep dividing as they're passing through different regions. They get progressively smaller calibers, but for as long as their position is steady, we're able to identify them and to follow them through their proper names. But at one point, smaller caliber arteries, as they keep dividing, they produce a set of so-called arterioles, smaller blood vessels, still maintaining the quality that arteries have, but visually they are much smaller, irregular in their appearance, and therefore they're not subject to further studies of anatomy. We do not necessarily follow them, see them, name them. We just understand that they must be positioned between larger regional arteries and capillary bed. So artery, after giving rise to multiple named smaller size arteries, following them until they divide to the level of arterioles. Basically, arterioles finally take us to the level of capillaries, both pulmonary or systemic capillaries within the rest of the body. Capillaries, through their thin and permeable wall, allow exchange of gases, exchange of nutrients, waste products, water-soluble vitamins, minerals, you name it. And of course, blood starts moving away from what we consider capillary bed within any given tissue. Blood, which is just about to emerge out of capillaries, as you can see here, kind of looks like illustrated as it changes the color. From bright red, it turns blue, which means at the arteriolar end of capillary, we had the blood enriched with oxygen. We delivered the oxygen to tissues, and on our way out from the capillary bed, blood started collecting and assembling more carbon dioxide. And that is why this change of the color has been illustrated. So once we do gas exchange and we move towards opposite end of capillary bed, basically we're entering system of very small, practically macroscopically invisible veins. We call them venules, and then venules are interposed in between the venous end of capillary bed and larger systemic veins that we identify, that we see at their standard location, and for most of them, we're also giving them proper name, so they become part of our studies of human anatomy. So to recap this in the shortest possible form, blood goes from heart into artery, which keeps dividing, giving us, at one point, arterioles, which also keep dividing and take us to the level of capillaries. This is called arterial end of capillary bed. Gas exchange takes place, and the blood is now reaching the opposite end, which is the venous end of the capillary bed, which is drained by smaller venules, which merge, and as they merge, they finally produce larger, macroscopically visible veins, and as a result of that, we're seeing their further merger until they form, as this diagram implies, two largest veins, one from the upper, one from the lower part of the body, and they were named as superior and inferior vena cava, respectively. I believe that this is pretty much a good chunk of information which will enable you to continue with your studies without any interruption. And there are a couple of extra diagrams which we are to see, but not really to spend as much time as what we have spent on the previous two diagrams. This is cross-section through artery and it is captured under 200 times magnification microscope. So we can see here the lumen, the cavity through which blood flows. Dark red is internal layer, tunica interna. Then the thickest part of the artery is obviously its muscular layer, followed by outermost layer, which is tunica externa, another ring of darker red colored structure. What surrounds artery on this picture is practically loose connective tissue, which is typically seen next to artery. At the lower part of the screen, you're now seeing 600 times magnification as the blood moves through a capillary. Please take a look and notice how big or how small are the red blood cells that you can actually see on this photograph. Their diameter is somewhere 6 to 7 micrometers, which means that size of capillary lumen is slightly larger just to make sure that red blood cells could move through capillary bed without being forced to stop or accidentally obstructing part of the capillary bed. To make sure that we are using proper and good quality terminology, we just need to expand things a little bit further. This diagram, very simply, is trying to illustrate what's happening with the heart during its contraction or systole, and then during its relaxation or diastole. We refer with these two terms, practically systole is contraction of a heart muscle, but more affecting ventricles than atria. Diastole, as the word, is also used to define what's happening currently with ventricles rather than to apply this entirely through both atria and both ventricles. So we use it quite loosely. However, atria and ventricles do have their separate systole and diastole, but it is more common that we're thinking about systole and diastole referring to activities of the larger two chambers, namely the ventricles. So what happens, we will see it later in physiology. There is a certain volume of blood which is ejected from a ventricle, and it is entering blood vessel, which is already filled up with blood. And we know that we cannot force the blood to be squished. It would require enormous force, and heart certainly doesn't have it. So as the extra 100-ish milliliters of blood are coming out of ventricle into aorta, obviously you can see here those multiple arrows that are pointing out to certain ability of aorta to extend its size, to expand, to dilate in order to absorb extra volume of blood. Arteries that are so close to the heart, including, of course, aorta, are considered to be elastic arteries. So in their walls, there are many more elastic fibers, allowing for this kind of elasticity in order to absorb whatever volume of blood comes out of ventricles. Further away we go, elasticity is on decline, but it is resulting in increased number of muscle fibers, creating the middle coat, tunica muscularis. So it is easy to understand what happens to blood during ventricular systole and the blood being ejected. Now let's try to understand what's going on during ventricular diastole. The elasticity of the aorta is now trying to reduce the size of it, bringing it back to what originally it has been. And that kind of elasticity recoil actually produces a bit of extra pressure, forcing blood to keep moving further away from the heart. Naturally, you're going to ask yourself a question: why blood doesn't go backwards into ventricle? Well, you can see on both upper and lower diagrams that there is some kind of physical barrier which prevents blood from being reversed backwards into ventricles. These structures are known as valves, and they regulate and ensure that the blood flow between ventricle and aorta, in this case left-sided ventricle, or on the right side, right ventricle and pulmonary trunk, that the blood is regulated and it flows from ventricle into aorta or pulmonary trunk, respectively. Once we pass those largest, closest to heart blood vessels with lots of elastic fibers, we would be entering into smaller caliber arteries section and arterioles, where elastic fibers are not predominantly present, but rather they're replaced with more muscle fibers, so that smaller arteries and arterioles could produce a pressure in their own and force the blood to keep moving away from the heart. Of course, capillaries are still a very interesting part of our studies. We cannot disregard the fact that capillaries, practically it's estimated that overall length of all capillaries in the human body is exceeding 300,000 kilometers, which again, if you try to compare it, it's 7 times around Earth, or a distance which is between Earth and the Moon. And definitely you can say it's quite impressive length of these valuable capillaries. Do they always have to allow traffic of blood to be constant? The answer is plain and simple, it's no. Occasionally, tissues have a higher metabolic demands, therefore insisting of capillary beds receiving sufficient amount of blood that is good to feed the tissue during its peak metabolic activities. But then comes the time when a certain part of the body is not utilized or used to its maximal, but rather 10% or 20% of its capacity. Then the blood becomes redirected. And that is what this picture illustrates, that we're seeing on the left side of the screen part of the capillary bed. Here's arterial, which feeds blood into arteriolar end of the capillary bed. Blood flows freely through it, enters the venous end of the capillary bed until it finally becomes drained into a smaller caliber venule. However, you see that there are a couple of smooth muscle fibers that are also part of this diagram that are wrapped around the capillary wall. What they do, they are practically functioning as sphincters. On the left-hand side, these sphincters are fully relaxed, enabling capillary bed to get filled with blood maximally. However, if the tissue is not at the peak of its metabolic demand, basically capillaries would still allow flow of blood through it, but with a reduced amount of blood. As the tissue doesn't need more, it needs less oxygen. Tissue doesn't need more nutrients, it requires less of them. Therefore, these pre-capillary sphincters, as they are contracted, basically they exclude part of the capillary bed from active circulation and ensures that there is a thorough, fair channel which practically continues through the capillary bed, enabling some blood circulation going from arterial into parts of capillary bed, being drained out of it, and then into venule and taken away back towards heart. That is explaining more physiologically functioning of the capillary bed rather than insisting on some kind of detailed anatomical recognition. But it is good to understand how this mechanism could affect the blood flow through capillary bed during peak hours of most intense maximal metabolic demands that the tissue may have on the cardiovascular system versus relative quiet times when the need for fresh blood, oxygen, and nutrients is quite reduced. Speaking of capillaries, you also need to know that not all of them are uniquely the same. There are 3 different types of capillaries which human body possesses, and they're all illustrated here: continuous, fenestrated, and sinusoid capillaries. Even if you do not delve any deeper into meaning of these terms, just glancing through these 3 different segments of this illustration, you will notice that there is physical difference what these capillaries look like. Continuous capillaries, obviously the word refers to some kind of continuity, is actually the most common type of capillary seen within a human body. Endothelial cells are very closely connected to each other. However, they're still leaving a little bit of a space between them, enabling the smallest size molecules to easily pass between endothelial cells, practically allowing exchange of gases, exchange of nutrients and waste products in any part of the body which is supplied by fresh blood. On the other side, we have fenestrated capillaries. The term fenestra, which means window, obviously offers additional openings not only between endothelial cells, but as illustrated here, through the endothelial cells. Fenestrations, or pores, actually are allowing more significant filtration to take place across the wall of fenestrated capillaries, such as what is capillaries of kidneys. We know that liquid part of our metabolism becomes purified after blood runs through the kidneys, which try to eliminate as many liquid dissolved byproducts of our metabolism and to secrete them into urine, which will be later eliminated from the body. The third type is called the sinusoid capillaries. And right away you can see that there are huge openings on the walls of sinusoid capillaries. Those type of capillaries are seen within liver, within spleen, and practically what they do, they really slow blood down massively. You will probably understand it when we use the example of liver much easier. Blood comes from our digestive tube, carrying all the different nutrients that we have absorbed across the wall of our gut into liver, which needs to do a number of different chemical reactions with the substances being absorbed. Some of them are not necessarily needed for the body or could have potentially even some kind of light toxic or any kind of non-benefiting to our body effect. They need to be eliminated. So how do we separate blood carrying potentially toxic substance and eliminate toxic substance away from it? By slowing it down and enabling liver to work very meticulously on content which has been brought into it by cardiovascular system. Spleen also slows the blood down, this time with one specific aim, to try to eliminate old, frail red blood cells from circulation. They would not be able to physically withstand the pressure of going from capillaries into large sinus-like spaces and then to be squished back into capillary, as this would be quite physical experience for them. So if they're old and worn out, the membrane of red blood cell will simply burst, and that's why spleen is sometimes more colloquially referred to being a graveyard of old red blood cells. On this picture, basically the emphasis is again to see what's happening within a vein. If we cut the vein open and we use frontal plane to cut perhaps 5 to 10 centimeter long segment of a vein, and then we make another transverse plane cut, basically we can see what the valve actually looks like. It is best defined as something that looks like a pocket being attached to the tunica intima. And when the pocket, this space here, or this one here, becomes filled up with blood, it simply pushes cusps of a valve closer to each other until they fully close and obstruct and prevent more reversed retrograde flow of blood through the vein. I believe that with the longitudinal cut and the lower picture, basically you can see even better why I compared the valves to some kind of attached pockets that are attached to the main wall of a vein. These pockets get filled up with blood, forcing the cusps of a valve to come closer to each other until they fully and completely close the vein, making sure that the blood can only flow one direction, from distal to proximal direction, or closer and closer to the heart. Veins of the lower limb are particularly prone to have issues with gravity. Gravitational force is constant. We cannot avoid it unless we're in the outer space. So as long as we're in a perpendicular position, basically the blood tends to pool in the lowest parts of the body. And in some professions, in people who unfortunately have to spend long hours standing while they perform their work, typically at one point in their life, they may start developing some issues, some problems about varicosity, unnatural dilation of these veins, and of course, improperly functioning valves. If they work functionally and properly, basically valves under pressure of the blood will be forced to open, blood will advance further up along the vein, perhaps forcing another valve to open, and the blood keeps moving closer to the heart. When blood eventually wants to go down, as it happens on the diagram 2, due to contraction of these two muscles and their compression along the wall of a vein, blood really keeps going in opposite direction, but that movement shuts the valve closed and prevents blood from being moved further backwards. This diagram is not only referring to value of valve, but rather also tells us about interesting and important addition to cardiovascular system, that contraction of our muscles actually is helping veins, acting as some kind of external pump. Because you see on the diagram 2 what happens upon contraction of these two unknown muscles. Part of the blood contained within a vein wants to go down, but it forces the valve to close, while contraction of the muscle still continues and moves and pushes further up and away the blood, forcing another valve proximal to contraction of the muscle to remain open and to allow blood flow. When these two muscles completed their action, they relax naturally. Blood also naturally wants to go down. However, it is going to reach this physical obstacle, the valve, which will shut close and prevent more significant retrograde movement of blood and more significant accumulation of blood in the lowest parts of our body. At this point, we would come to next diagram, obviously taking us into the direction of starting our studies of the heart. I will stop here for a moment just to allow these videos to be as short as they can be and to use this as ending point for first section and a starting position for part number 2 that will be recorded and posted. Basically, we just want to make sure that we understand properly where the location of the heart is, together with its protective membranes that are surrounding it, illustrated by a black dotted line. They are called pericardia, and the same thing that we see on the lower part of the diagram just being illustrated in frontal plane as opposed to transverse plane, the upper diagram illustrates. So I'm going to stop here, and as always, thank you for watching this video, part 1 of the cardiovascular system anatomy, which addressed the principal setup of the system, and we spent a little bit of time talking about arteries, veins, capillaries, and their most obvious and most important functions. Stay tuned, part number 2 is coming soon.